Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall look at receptors for cell signaling and we'll focus on the ligand-gated ion channels. In the last video, we talked about the basics of cell signaling and how our 30 trillion cells communicate with one another by this process called cell signaling, and we also mentioned the different types of cell signaling that we find in our body. Just a quick recap, in cell signaling, the signal binds a receptor, and when the signal binds the receptor changes shape, and when it changes shape, that triggers a signaling pathway where the receptor will affect other proteins, which in turn will affect even more proteins in the cell. The end result of this is various cellular proteins in the cell will be affected. Either their functions may be increased or decreased, the amounts of the proteins in the cell may be increased or decreased, and even the location of the protein may be altered. So as you know, proteins do almost everything for cells. So once we start changing this amount of proteins or functions of proteins or locations of proteins, that's going to lead to a change in cellular activity, a biological outcome such as changes in metabolism, changes in cell division, survival or apoptosis, and it may even make the cell differentiate into some other type of cell. Although there are many signals found in the human body, the number of receptors are limited. So far we see only a few families of receptors. So in the next few videos we shall talk about some of the more important families of receptors that we see in our cell, and that includes the ligand-gated ion channel receptor, which is also called the ion channel link receptor, as well as the inotropic receptor. Then we'll look at the most common type, which is called G protein coupled receptor. And the G protein that is associated with this receptor could be the G-alpha S, G-alpha I, or G-alpha Q that we'll talk about in future videos. And then we have the enzyme link receptors, where the enzyme activity could be intrinsic to the receptor, or it could be associated. The enzyme activity could be associated with the receptor. So these three here are surface receptors. And then we'll talk about the intracellular receptors. These are receptors found inside the cell rather than on the surface. We have covered these receptors in our previous videos. So for example, the nicotinic acetylcholine receptor is an example of a ligand-gated ion channel receptor where the signal is acetylcholine. An example of a G protein coupled receptor that uses or is associated with G-alpha S protein is the glucagon receptor where the signal is glucagon. An example of a GPCR that is connected to the G-alpha I protein is the acetylcholine receptor type M2 as well as M4, where the signal is acetylcholine, while a GPCR associated with another G protein called G-alpha Q is also an acetylcholine receptor of a different type, the M1 and the M3 type, and there too the signal once again is acetylcholine. The enzyme link receptors, we have one type where the enzyme activity is intrinsic to the receptor. We did talk about this as an example, the RTK receptor, that was the insulin receptor as an example, and insulin there is the signal. And then we talked about the enzyme link receptor in a previous video that has associated enzyme activity. It doesn't have intrinsic activity, but it can recruit an enzyme to the receptor. And there we had mentioned the leptin receptor as an example, where the signal is leptin. And as far as intracellular receptor, we did talk about one of them, and that was the glucocorticoid receptor that responded to the signal called cortisol. So in this video, we are going to focus on the ligand-gated ion channel receptors. We'll look at a few examples of these receptors as well as the signals that they bind to. Let's first look at this nicotinic acetylcholine receptor where the signal is acetylcholine. So here we discussed this in a previous video. What we have is the neuromuscular junction, and the nerve here sends out a signal called acetylcholine, and this signal will bind to the nicotinic acetylcholine receptors present on the muscle cell, the plasma membrane of the muscle cell. That leads to a change in the receptor shape, and that causes sodium ion channel of the receptor to open, which results in depolarization and action potential. And that action potential travels through the plasma membrane of the muscle cell and causes the opening of calcium channels in the T-tubules, resulting in calcium entry into the cell. And that calcium entry into the cell is going to make more calcium channels open, and these calcium channels are found in the sarcoplasmic reticulum, and opening of these calcium channels releases a lot of calcium, which then causes the muscle to contract. So let's look a little bit more closely at this receptor. So if we look at the structure of this receptor, it's a surface receptor found on the plasma membrane. This receptor, the NACHR, has 5 subunits that are arranged around a central channel. And this channel in the middle here, this channel is an ion channel for sodium. So when the signal acetylcholine, that's your natural signal, even nicotine can bind to this receptor. So when the natural signal acetylcholine binds to the receptor at the 2 alpha subunits, the receptor shape changes. And when the receptor shape changes, there is a helix that is blocking the channel. That helix is moved away, and the ion channel opens very quickly within milliseconds of the signal acetylcholine binding to the receptor. So since we have a receptor where there is a signal binding, the signal here is the ligand, and that causes a channel that's associated with the receptor to open. That's why we call this ligand-gated ion channel. So these type of receptors have a portion on the receptor which binds the signal, and also a channel that allows ions to either enter or leave the cell. So in this case, the ion channel is more selective for sodium, since there is a higher concentration of sodium outside the cell. And remember, that's because of the sodium-potassium pumps, which are actively working in most cells. There's a higher concentration of sodium outside the cells. So once this channel opens, the sodium from outside the cell will start diffusing through this ion channel and will enter the cell. There's a little bit of potassium that might also exit, but mostly sodium enters the cell. So since sodium is positively charged, we are sending more positive charge into the cell. That's going to lead to a depolarization of the muscle cell, and that then leads to the action potential and all the other things we talked about earlier. Eventually calcium enters the muscle cell, and there's large amount of calcium inside the cytoplasm of the muscle cell is going to cause the muscle to contract. So that's the basic concept of a ligand-gated ion channel receptor. So the receptor that has a binding site for the ligand, for the signal, and has an ion channel that can open and close depending on whether the signal is bound or not. Let's look at another example of a ligand-gated ion channel, and this is the NMDA AMPA receptor. The signal for this receptor is the amino acid glutamate. So these two receptors, the AMPA as well as the NMDA, these are surface receptors. Now they coexist on the same membrane. So wherever there is an NMDA on the membrane, there is also going to be the AMPA receptor on the membrane. So NMDA stands for N-methyl D-aspartate, and AMPA stands for amino 3-hydroxyl 5-methyl isoxazolipinic acid. And these two receptors, as mentioned, coexist on the same membrane. Now, unlike the AMPA, the NMDA has a magnesium ion in the ion channel of the receptor. AMPA doesn't have that. The ion channel doesn't have a magnesium blocking it, but NMDA does have a magnesium ion that is blocking the ion channel. So when the signal glutamic acid binds to these receptors, so binds to the NMDA receptor as well as the AMPA receptor, the receptor shape changes. So the ion channel for the AMPA, the sodium ion channel, will open, and then sodium is going to enter from the outside, where it's present in higher concentrations, to the inside of the cell as the channel opens. However, sodium and or calcium cannot enter the NMDA channel, although it's open. And that's because of this blockage by magnesium. So cations cannot enter through NMDA, they can only enter through AMPA. So as more sodium enters AMPA, that leads to a depolarization of the membrane. There's more and more positive charge inside the cell. And this positive charge is now going to repel the magnesium ion from the NMDA channel. So the magnesium ion present here, which is also positively charged, is going to be repelled, and the magnesium ion comes out of the channel. So with the magnesium blockage removed, now we see sodium, and this channel also allows calcium. So sodium ions and calcium ions from the outside of the cell will now start entering into the neuron. So now more sodium means depolarization becomes more stronger. So this magnesium is kept away. Calcium and sodium keeps on entering. And as calcium enters the neuron, the calcium, as you know, calcium is a very important intracellular messenger. It affects the functions of many proteins. And what happens, it goes and opens up more and more AMPA channels. So the calcium here will cause more AMPA channels to open. It also increases AMPA receptor transport to the plasma membrane, and also increases the release of the signal glutamic acid. So all of these processes will enhance the synapse, and that leads to a long-term potentiation. This is a physiological process that is very important for learning as well as other neuronal functions. So So this AMPA, NMDA channels play a very important role in learning as far as the brain is concerned. Let's look at another example of ligand-gated ion channel, and in this case we'll look at the glycine receptor, where the signal is the amino acid glycine. So the glycine receptor also has several subunits that are arranged around a central channel, but in this case the channel is specific for chloride ions. So when the signal glycine binds to the receptor, so when this glycine binds to the receptor shape changes, conformation changes, that causes the chloride channel to open. Now chloride ions, which are present in higher concentration outside, will now enter the neuron. So now we are bringing in more negative charge, not positive charge. So in this case, the membrane is hyperpolarized, more negative charge into the cell. And in this case, the neuron firing is inhibited. A similar mechanism is also seen with the GABA-A receptors that we find in the brain and elsewhere. So hopefully these examples will give you a good idea of the structure and functioning of these ligand-gated ion channel receptors that are also called ion channel link receptors or inotropic receptors. In the next video, we shall look at the GPCRs. So until the next video, once again, thank you very much for supporting this channel. Have a wonderful, wonderful day, and we will see you soon.